Okay, we are going to continue and hopefully wrap up our discussion of cell membranes by first taking an overarching look at their overall structure. We've looked at details up to now, and then we're going to go into how their structure enables them to do the various jobs that they do. Okay, so the parts of the cell membrane, which we've already discussed, are the phospholipids, which is kind of the main part, the foundation part of the cell membrane. It's the bulk of it. It's like having a brick wall and made of bricks, and the bricks, in this case, are the phospholipids. But oftentimes bricks aren't all you have in a wall. You might have a window or a door or a hook or anything hanging on a wall, a painting, something. So there are other things that are part of the wall that we need to talk about. Now, those things that are part of the wall, part of that phospholipid foundation, are proteins. These proteins are embedded among the phospholipids. Often the example is something like raisin bread, okay, where you have this slice of bread that is the bread is your phospholipid foundation, but there might be raisins sprinkled through it. Those are like the proteins in a cell membrane. And these proteins can have many different jobs depending on what protein they are. Some of the proteins act as little channels, little tunnels that transport materials from one side of the membrane to the other, from in to out or out to in. Some can be pumps that actually force molecules to move in one direction to another, either into the cell or out of a cell. Some can act as identification markers, ID markers, if you will, that allow a cell to be recognized for what it is, a cell belonging to me or to you. And so for example, your immune system can recognize a cell belongs to you and not attack it. Whereas if a foreign invader comes into your body, a bacterial cell, it wouldn't have the proper ID marker on it, so your immune system could take care of it. Proteins embedded in cell membranes can also act as receptors. They have special little shapes that allow them to receive certain molecules from the environment, bloodstream, whatever, and that can cause them to do certain activities. Okay? So, this idea of a phospholipid bilayer embedded with little proteins and other things that do work is called the fluid mosaic model. Okay, this idea, golly, I'm at a loss for when they came up with. Sometime in the mid part of the last century, it was determined that this was basically how cell membranes were built. There were many phospholipids, and they aren't stuck together. These phospholipids aren't stuck to each other. They can kind of slide around each other, kind of like a soap bubble. If you blow a soap bubble, that soap isn't stationary. You can kind of watch the soap and the soap bubble fluidly move around. The phospholipids are the same way, hence it is fluid. But it's also a mosaic, meaning it's made up of bunches of little parts, whether it be phospholipids or these big globby proteins that are embedded in it. So here would be a big glob of protein. Here's one, here's one. Some of them act as channels, like this one. You can see that it's like a little tunnel and something could pass through it. This one is a receptor, but it's not all that different from an identification marker that's a protein with a little special funky identification tag stuck to it. It's usually these are made out of carbohydrate parts, while this is a protein piece down here. You don't need to get all bogged down in that. Some proteins act like pumps. Some act like enzymes. There's all kinds of things, okay? But generally speaking, that idea of the fluid mosaic model is how we talk about cell membranes being put together. Now, as we've already mentioned, there are many types of proteins embedded in cell membranes. cell identification markers are one important type for you to know about. So if this is my phospholipid bilayer, there's my phosphate heads and my polar tails. Each of these is a phospholipid. Okay, if it's a bilayer, I've got another set that goes opposite them. And embedded across these might be a glob of protein. Okay, and you can see it's embedded in this phospholipid bed. Now, sticking off of this particular protein might be an identification marker, some kind of uniquely shaped, usually some type of sugar. It's usually not a super long polysaccharide, but it's usually bigger than a disaccharide. It's a sugar molecule, kind of a big sugar molecule here. And it's got a unique shape. It would be unique to you or to me or whoever. This is a cell identification marker, and your cells all have these particular markers that let your immune system realize that, hey, this belongs to you, okay? And not to attack it. Now, receptors would look a whole lot like that. Here's my phospholipid bilayer one more time. Here's another protein glob embedded in the bilayer. Whoops, sorry. And, okay, and instead of just being a weird shape thing, this time this is going to have a particular shape that can receive a molecule. So if there's a hormone, for example, traveling in your bloodstream that needs to tell a cell to do something, maybe it's a growth hormone, it needs to tell the cell to grow, okay? If it is shaped correctly, it will fit this receptor, the receptor will cause the protein to do something inside the cell here. Maybe it will cause some chemical reactions to occur on the other side. And some action inside the cell is caused to happen because this guy bound to the outside of the cell in this receptor. Other molecules, maybe different hormones that are not related to what this thing is supposed to get, might go by, and this one wouldn't fit. Okay, it will only fit the one particular molecule it is designed for. Okay, so those are receptors that can receive molecules and allow the cell to do work, respond to that receiving of that molecule. Okay, there are also transport proteins. Okay, and these are the most important ones for the remainder of our discussion. You just kind of need to be aware that receptors and ID markers exist. These guys, however, we need to really talk about what they do. Channels and pumps. Again, these are both different types of proteins that could be embedded in a cell membrane. But channels are going to act like little tunnels, if you will, and molecules will be allowed to pass from one side to get from outside to inside the cell. I finished drawing my phospholipid bilayer, but you get the idea. Okay? And not just anything can get through this special little doorway. Only a particular molecule that has the precise shape that is needed to get through that little tunnel will be allowed to pass. Okay? Pumps are going to force things to pass. Maybe even when they don't, quote, want to, unquote. Now we'll talk about what I mean by that very shortly. So, for example, here you go. Here's phospholipid bilayers, and each of these represents a channel, okay? This is one part of what makes a membrane semi-permeable. Some things are allowed to pass, but not all things. We've already talked about that. Sometimes really small things like an oxygen molecule that's nonpolar can squeak through, but we've talked about other things that can't squeak through, okay, even though we might need them to. Like, for example, this sugar molecule or this amino acid. If this is the outside of the cell and this is the inside of the cell, maybe this needs to go into the cell to provide nutrition. Maybe this amino acid needed to get through to allow proteins to be made. We got to have amino acids to make proteins. Maybe some water needed to get through. So we need to provide a channel for that. Maybe some salt needed to be excreted. Maybe we had too much salt in the cell. Maybe we needed to get that out. Maybe we need to get a waste product, ammonia, out of the cell. And so we allow that to pass through a particular channel. The point is that we aren't letting just anything pass. If we take away these channels, amino acids will not go through. And other things can't go through this channel, only this particular amino acid. So we can control very tightly what goes in and out of the cell by controlling what kind of protein channels are placed in the cell's membrane. Okay? That makes it semi-permeable. Control is maintained. Okay? Specific channels allow specific material to cross the cell membrane. Now, in general, there are two main types of transport. One of them is passive and one of them is active. This is broad overarching categories. We are going to talk about passive transport first. All right? In passive transport, the cell does not need to use energy, does not need to use its own energy to do anything. Passive transport is when molecules will move from outside to inside or in to out, whichever direction you're talking about, without the cell having to do anything. Okay? It just occurs. Why does this movement of molecules occur? Because the molecules are moving with their concentration gradient. And you might say to me, but Ms. Coleman, what the heck is a concentration gradient? And I say to you, give me a minute and we'll talk about it. This passive transport happens because of one thing, okay? Molecules are always moving. Molecules have molecular motion. They have kinetic energy. They're always moving. And it is just because molecules are always moving, the air molecules around me are moving right now, random motion. That random motion will end up carrying certain molecules across the cell membrane if they have a way to get through the cell membrane, okay? An appropriate opening, a protein channel, a channel made out of protein, must be present usually to allow molecules to pass. Not always. Remember, if something is small and nonpolar enough, it may be able to pass through the phospholipids between them. But in general, we got to have a channel for most things. Now, there are two main types of passive transport, okay? Remember, we said there's passive and there's active transport. There are two subgroups of passive transport. One of them is called diffusion. The other is osmosis. Let's see what those are about, okay? Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. What that means is from an area where there is a lot of them together to an area where there is not so many of them. So, let's say I have this container, and this is a semi-permeable barrier here, so things can get through it. And I have a bunch of molecules on this side, but I have no molecules over here. The random molecular motion of these molecules is they're going to be moving around. They are highly concentrated on this side. There is only a low concentration, like none, on this side. There's lots over here, none over here. Their random motion, as long as they can get through this barrier that is semi-permeable, will carry molecules randomly to this side from over here. Maybe this guy goes, maybe this guy goes, until they are evenly dispersed. Until they're equal amounts on left and right. Now, just because I then, after time has passed, had equal amounts on this side and the other does not mean the molecular motion stops. It just means we couldn't perceive a difference anymore, because there would be equal amounts on each side, and the molecular motion would just be carrying them back and forth, and we can't really perceive that happening because they're in equal amounts and it all looks kind of the same to us. Think about food coloring. If I put a bunch, here's my semi-permeable barrier, if I put a bunch of food coloring molecules over here, I can see it's really blue on this side, but I can't, but there's none over here. But as those molecules begin to move, I'm going to see it's going to get more blue, so now it's going to be blue on both sides, and the molecules will still go back and forth, but I can't really perceive it anymore because they're both the same color and it's not going to be visible. So, again, let's not get bogged down. Diffusion is the movement of molecules from an area of high concentration to an area of low concentration. And they do that, they never stop moving, but we stop being able to perceive motion once they reach equilibrium, meaning once there are equal amounts of molecules on either side. So then that diffusion stops as far as we're concerned. Now, what this means when they are going from high to low, they are going with the concentration gradient. They are going with the gradient from high to low. Now, depending on the molecule, they may or may not require a channel protein. Remember, something like oxygen, which is small and nonpolar, that can squeeze between the phospholipids. But some things, like a big old sugar molecule, is going to require a channel protein be there. But the channel is nothing more than a door, an open door for my sugar molecules that will go back and forth with their concentration gradient. So, facilitated diffusion, this word, facilitate, means to help. Facilitated diffusion then is just diffusion that has to occur through a little helper channel. Okay, that's all that is. Now, osmosis is simply diffusion. It is what we just learned about. But it is the movement of water molecules from an area of high concentration to an area of low concentration. Again, this is with the flow of the concentration gradient from high to low. The only difference between this and diffusion is that we are specifically addressing water molecules now. Okay? Now, active transport, on the other hand, our other big category, it occurs when the cell has to use energy to make this motion occur. It's not going to just happen because the molecules are moving. We are going to have to put energy into pulling some molecules because they are now going to be forced to go against their concentration gradient. What does that mean? We are going to move molecules from an area of low concentration to high, which is going to require us to go against their normal flow. Normally, molecules want to go from high to low, from this side to this side. What we are going to ask these molecules to go is to go in that direction, low to high. This is going to require a pump, an energy input from the cell in order to do that. Why would we ever want to do this anyway? Because, imagine, cells, here's a cell, it's got a fair amount of sugar molecules in it. Let's say that's a sugar molecule, sugar, 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 sugar. And we may not have that much concentrated sugar in our bloodstream, but does the cell want more sugar? Always. It's a nutrient. So we're going to pump that sugar in against its concentration gradient. Even though we've already got lots in here, we're going to bring in even more. Okay? Now, we are going to stop there, and then I will pick this up with one more little quick podcast talking about certain types of osmosis and how osmosis works. Okay? That's it for now. Take it easy, and I will catch you in the next podcast.